And my lab is largely interested in looking at antibiotic-producing bacteria, both for the discovery of new antibiotics and then looking at biosynthetic pathways on how these antibiotics are made in these bacteria, and then doing genetic manipulations to coerce bacteria into making analogs. And one of the main projects that we work on has to do with an antibiotic that's used as an animal feed additive. And when Greg came, I guess it was 2009, January 2009, so it's quite a while, looking for a project, this seemed like the first thing to toss him onto was, okay, let's get your hands wet in the lab. And we needed a bunch of this stuff isolated for Neil Goebel to work on as part of his thesis project. He needed starting material from this peptide antibiotic that came to us in I think it's a 20-kilo bag mixed with talcum powder and cornstarch that they feed to these chickens. And so it was a bit of a nightmare, but that was his baptism by fire in the lab. But from that, a really interesting project came out of it, led to some interesting new compounds that are found in this feed additive. And so it was, again, following the data, the information, that led Greg to this project. And then he also took on another project having to do with screening some bacteria for novel bioactive compounds in collaboration with the Hai Zhang, who's one of the postdocs in the lab. But it was a real pleasure having Greg work in the lab. I think that the BRR students that we've worked with in the past really bring broad training and broad interests into these research projects, these thesis projects. And I think that's why Carrie and I always welcomed, and others, I guess James also had BRR students, have welcomed them into the labs, because they're really a pleasure to work with. Very interdisciplinary, very creative, and risk-taking. So Greg turned out to be just like the others, and it's been a real pleasure to work with over these past year and a half. Wonderful. Anyway, so let's let Greg tell us a little bit about the projects now.
Thank you for the elaborate introduction. As Dr. Zabriskie said, this is my lab mates, and Dr. Zabriskie is my principal investigator. The title of my presentation today is Discovery and Isolation of Bioactive Natural Products from Actinomycete Bacteria, which Dr. Zabriskie gave us a very good overview of, and I'll probably touch on again. So let's begin by starting. I think you went to screen saver. Back. All right, let's go back. Introduction. All right. So I began from the beginning of the project looking at drug discovery. There are two paths most pharmaceutical companies can take. There's screening through synthetic libraries, and there's screening through natural products. And the biggest beneficial factor of natural products are they evolved specifically to interact with biological receptors and other bacteria. We go to find these natural products that are produced by bacteria in unique environments to hopefully find new and novel compounds. We try and find diverse isolates of bacteria, and we try and get locations that have been sort of untapped. This is an overview of my work, which Dr. Zabriskie elaborated on, beginning with the screening of our actinomycete strains from Indonesia for Staphylococcus aureus inhibition. We're focusing only on Staphylococcus aureus. And then purification of indorecidin, the potent antibiotic used for our commercial feed additive, from enerdin, the big sac which he was talking about. And last but not least, the enerdin fraction, which we found to have bioactivity, which I went on to isolate and purify. What are actinomycetes? The order Actinomycetales are gram-positive, aerobic, chemoorganotrophic bacteria. Chemoorganic trophic meaning they derive their chemical energy from the breaking of bonds, and they utilize hydrogen and electrons from organic sources. The order Actinomycetales was first wrongly classified as a fungus. But later it is elucidated that they are mycelium producing, and they just have this diverse metabolic activity. They have up to 30 gene clusters which code for alternative compounds. These actinobacteria natural products are responsible for multitudes of antibiotics we use today. There's an in vogue genus called Streptomyces, which has provided us with antibacterials, antifungals, anti-cancers, antiparasitics, statin drugs for blood pressure, and immunosuppressants. So a wide array of uses just from this one genus of bacteria. This is a gram stain of Actinomycete Bacteria. As you can see, it's very filamentous. It is purple, which is characterized by a gram-positive result. And yeah. Collection of the organisms. These rare non-streptomyces, actinomycete is what we were looking for, something that other researchers haven't already delved into. We pulled these isolates from an exclusive blackwater ecosystem in southern Indonesia. Blackwater ecosystem is characterized by a lot of dissolved organic matter in the water. There's horizontal ground flow, so there's a lot of sediment and things from the surrounding environments that are going to come into the water. Come on. Okay. It's very acidic and has a tea cola appearance, which is why we call it black water. It's very dark. Many thanks to the Indonesian Center of Biodiversity and Biotechnology, which collected our microorganisms. I have a map up here to show you. The river is in southern Kalimantan. Hopefully I said that correctly. And it's this is the one area that, of all of Indonesia, has been more conserved rather than the rest of the country. So being able to get our isolates from this area is very lucky. So complex medium fermentation. We transferred our bacteria from a simple media culture to put them into environments that would try and induce these secondary metabolite pathways. We used 6 unique complex mediums to do this, to promote the secondary metabolite production. And I went all the way from the beginning of drug discovery, where we ferment the bacteria and try and get them to produce this metabolite. We harvest what they have produced, and then we screen for them using a 96-well plate. This is the beginning of harvesting and isolating the compounds that actinomycetes created. This top left corner, you can see the simple media, just culture of the strain to have a reservoir of our bacteria. And then we moved on to inoculating these 6 different media wells. So one strain for these 6 different media wells. And there's about 8 milliliters in each well. And so after inoculation, we would let them grow for about 7 to 10 days. And at the end of it all, you could visibly see the difference of these metabolites in each well. So the bacteria are definitely producing something different in each environment. I went on to pulling the full complex media and the strain or whatever they've produced and transferring it over to a 25 milliliter flask, where I went on to crudely purify anything I could and pull out by solvent extraction in 2 steps to get as much of the gunk or organic matter as I could out of the the fermentation. And finally, I would end up in a 2 milliliter Eppendorf tube, the most crude purified metabolite I could get from each well. So from each well, I have my tube, and I used 2 different dilution factors. One at 50 milligrams per milliliter and another at 5 milligrams per milliliter to test different concentrations of the metabolites to see how potent or how effective they are. The 50 milligram per milliliter is more of just a general, yes, this produces something that can inhibit bacteria because it is such a large concentration. So it's either the yes or no factor. And then going down to the 5 milligram per milliliter concentration, looking at, okay, well, does this metabolite, is it potent enough to work at small concentrations to give us a good antibiotic? So a 96-well bioassay method was an improvised method used by Smith et al., published in 2006. Our improvised method employed this high concentration, 50 milligrams, and low concentration, 5 milligrams per milliliter assay. We marked our bioactivity to be a growth of less than 50% Staphylococcus aureus. We had a positive control of actinomycin, which is an antibiotic used against Staphylococcus aureus, and a serial dilution to see at what concentrations this antibiotic is potent, to refer back to our metabolites to see how to correlate the two. We had our negative control of Staphylococcus aureus and media to just have our benchmark turbidity of maximum growth. And we had our blank, which was just media to make sure that it was not contaminated. This is a 96-well plate map. This was my map that gave me the initial bioactive indications that these strains were producing metabolite to inhibit the growth of Staphylococcus aureus. At the top is my actinomycin control, positive control in red. Bottom right corner is my blanks, which you saw less than 0%, so I had no contamination in my media. And my negative controls were used for the maximum growth, as I said, correlating back to how much growth there is in each of these wells. As you can see, 8340 in our first complex media well at 30 milligrams per milliliter gave us a growth of 31%, 45%, and 53%, right at our cusp of bioactivity. Here is our strain 8352 in a different complex medium 6C at 50 milligrams per milliliter, giving us a lot better inhibition at 16%, 15% growth. So referring back to this point, after I got this bioactive indication that these strains can make something, I went on to ferment them in a large scale, about 250 milliliters for both strains. So instead of inoculating my 8 milliliter complex media strain, I inoculated this 250 milliliter flask. And this is what it looks like. This is my strain 8340 after it has been cultivated. And this time, for the large-scale fermentation, I went on to separate the mycelia and the culture broth to test the two different sources where this metabolite can be found. So I centrifuged it down at about 20 minutes at 3,000 RPMs and got a good separation and was able to test these two separate components, find where this metabolite is. So as I said, the ICBB8340 strain, I fermented in M2 media. It gave me this pastel pink culture broth. I found that there was no bioactivity at any concentration under the 50 milligram per milliliter, so it was not a good candidate against Staphylococcus aureus. I then went on to give both my strains, 8340 and 8352, to our postdoc, Dr. Dahai Zhang, to verify whether it was my fault that these bioactivities weren't coming up, or there's some other factor that I must have missed. His results came out to be there was no bioactivity in either of the strains under 10 milligrams per milliliter, so this was again not a good candidate against Staphylococcus aureus. Maybe for a different target, these metabolites could work better. So the conclusions of this part of my research is I went on to work a little bit more with the enerdin fraction that I found to be bioactive. So these metabolites were not effective at low concentrations, thus they were not powerful agents against Staphylococcus aureus, to reiterate. And Dr. Dahai Zhang carried out the screening of the remaining rare non-streptomyces that we got from Indonesia. We'll just wait a couple seconds for this to work. Sorry for the technical difficulties. Okay. So this is the second part of my thesis. Isolation of a new antibiotic from enerdin. As Dr. Zabriskie was saying, this is the enerdin side. that's been infused with starch and I don't know why it's doing this. Anyway, starch and some other substances that are used for the commercial feed additive, and it's for the poultry industry. Anyway, now that the slide's back up, endogen is a derived biomass from the bacteria Streptomyces fungicidicus. They are the bacteria that produce endurocidin, and there's about 5 to 8% derived weight of endurocidin. Now, thinking this is a pretty low percentage to have something in such a big 20-kilo bag. Streptomyces fungicidicus actually has a really hard time pumping up this endurocidin, which is why it's very not only expensive but sought after by chicken farmers. So endurocidin is a peptide antibiotic. There is no known mechanism of resistance as of today, and there hasn't been for many years. It is targeting gram-positive bacteria, but most emphasis put on its methicillin-resistant Staphylococcus aureus activity, and it's very effective at killing these resistant strain Staphylococcus aureus bacteria. This is endurocidin, the endurocidin compound. It is very large. Natural products range from small to large, and this is a very large natural product. As you can see, it has a very complex ring system. It is very rich in oxygen, and has many places for modification. Well, our grad student Neil Goebel, he works with modifying this endurocidin to either increase solubility or increase its potency. And as you can see, there's many different places for this modification to happen. This is my scheme for the purification of endurocidin. These are many parts, so you guys are going to have to bear with me. It begins with, at the top, we have our endogen sac. We take about 20 grams of it, and we just do a solvent extraction using phosphoric acid and acetone to pull whatever compounds we can into a liquid form. And from this crude endogen extract, we take some silica gel and mix it with the liquid solvent and dry it onto the silica gel. Thus it is adsorbed onto the silica gel, meaning the silica gel is here and it coats the outside of the silica gel instead of being absorbed into the silica gel. Once the silica gel and endogen is infused with each other, we can put them in a column and elute out the compounds connected to the silica gel with different solvents. And in increasing polarity, we began with THF, tetrahydrofuran, DCN dichloromethane, to 90% acetonitrile to 80% acetonitrile aqueous. So we knew at 80% acetonitrile is where our endurocidin would elute off. We can check this by TLC plate. Luckily, endurocidin is UV active, so as soon as you get your TLC plate and you put it under the black light, you can see whether it is positive for endurocidin or not. And so before we moved on to the next part of our purification, Neil Goebel thought it would be a good idea to find whether our endurocidin was actually in this 80% acetonitrile fraction by testing it on a disc diffusion bioassay. This is a disc diffusion bioassay where each disc has about 20 microliters of each of the elutants dried onto it. We have our blanks in the middle, and we had our known 80% acetonitrile here to give us inhibition. And here we had our 10% acetonitrile, which pulled off a little bit of the endurocidin, but not all of it, which is why it has a smaller ring of inhibition than our 80%. And what was the most surprising is why our THF and DCM were giving such a huge inhibition of the Staphylococcus lawn. So we went back to the drawing board and marked our THF elution as bioactive and put it aside. We went on to purify the 80% acetonitrile elution by infusing again with the ion exchange column X8016. where we went on to have our semi-purified endurocidin by decreasing polarity of isopropyl alcohol, where we knew 70% isopropyl alcohol would pull off our endurocidin the best. 30% isopropyl gave us a little bit of it, but not as much as 70% isopropyl alcohol. We yielded in total about 16.1 milligrams from our 20-gram energen, which is about 0.8%. 1% of the full 5 to 8% of endurocidin in the bag. So pretty good yield. We were happy with what we got. Going back to the THF fraction that we found to have bioactivity, this THF fraction was again loaded onto a silica gel column, and we looted off with 2 different solvents this time. 1% methanol in dichloromethane and 5% methanol in dichloromethane. We utilized TLC to be able to pool different compounds, compound fractions into whole reservoirs. So these separate compounds were separated into the different fractions. We tested again with a disc diffusion assay, as you guys saw earlier, but this time it was indicated by diazosyl blue tetrazolium, which in live bacteria is reduced in the mitochondria to produce this vibrant violet color, which you'll see here. And so with the vibrant violet color, that means these cells are alive. And if you look at the actinomycete control, we have a big ring of inhibition where no violet was produced. And not only at the actinomycete control, but in our fractions 37 through 41, housed our bioactive compound. From here, we can use HPLC, which is high pressure liquid chromatography, which separates mostly by charge and size of compounds, and acts as a silica gel, except much more attuned to seeing what your compound is and being able to use the computer to elute off exactly what compounds you want. And so on the left is a 96-well plate, which the HPLC was eluting our compounds into. This is the initial chromatogram that we saw with this 37 through 41 fraction. As you see right here in the middle, this very unique UV absorption, which we thought, okay, well, this must be a really interesting hit. So we went on to use our HPLC and collect our compounds in this 96-well plate, and again do another bioassay using the Smith et al. Methods published in 2006. Same positive control, our serial dilution of actinomycin, our negative control again, Staphylococcus aureus and media, allowing that to grow to its maximum yield, and our blank was just our media. We measured the turbidity at time 0 and time 17 hours to see what happened with the growth and what compound is active. And so the HPLC, in a zigzag manner, eluted out the compounds up to 72nd well. Top, again, the actinomycin control, or blanks, and the negative control. So our results produced very good inhibition at some points. We see this is the percent growth, so the lower the growth, the more inhibition. And this was the well number, again from 0 all the way to 72. We see here there's distinct different compounds between the fractions. So we have more than one compound that is causing our bioactivity. So from there we want to purify even more so we can find what is giving us this inhibition. We marked our chromatogram of the different compounds. We had compounds A through H, but as I said, D and E provided us with the most interesting UV absorption. So I was able to give these compounds to our grad student, Neil Goebel, and he went on to characterize it and determine its structure. Turned out compounds D and E, which were most interesting, were actually a keto-conjugated unsaturated fatty acid. This is acts as a detergent by incorporating itself into cell walls and causing cell death, rather than antibiotic or peptide antibiotic, which we hoped that it would be. So our conclusions. The endurocidin purification was definitely maximized. We had great 16.1 milligram yield, which Neil Goebel can go on and make his modifications to, as I said, improve solubility and try and maintain its bioactivity or even increase its bioactivity. Our bioactive THF fraction contained that unsaturated fatty acid, and we concluded that all compounds from A to H were just analogs of this fatty acid. So this is a common nuisance for natural product drug discovery. It inhibits the bacterial growth, but it does not make an ideal antibiotic because, as I said, it acts as a detergent. To further reflect on my work, natural products are essential for battle over drug resistance. As I said in the beginning, natural products were made by biology to interact with biology. We must look for new fermentation mediums to hopefully induce these secondary metabolite pathways and new high-throughput methods, such as maybe separating the cultivation part of the bacteria and the metabolites and separating with the screening. So having two different labs do the work instead of my work where I did from beginning to end. I would like to acknowledge the Subsurface Biosphere Initiative Summer Undergraduate Internship Program for allowing me to have the time and money to work through this presentation, to work through my work over the summer. I'd also like to thank Intervet, which was before known as Sharing Plow Animal Health, for our gift of Enerdin that we've been working with. I would like to thank Dr. Mark Zabriskie and his lab for taking me under their wing and allowing me to do my work and understand a lot more than I would if I didn't do this research. This research has really opened my eyes to academia and how we must push forward for new drugs. I would also like to thank the BioResource Research Department for the great counseling I've had over the years and allowing me to present this thesis. And I'd like to thank our institution, Oregon State University. So, thank you. Thank you all for coming. I really appreciate it. I really appreciate it.
Questions from the audience for Greg?
Yes.
In your first part of the work that you presented,
Yes.
You said that the organism that you collected came from the hydrotritis, blackwater,
Yes.
Low pH, and you had several broths and media that you cultured and grew them in.
Yes.
What was the pH of
The
those?
pH, they were acidic pH, so I had to adjust the pH of all my complex medium to be around, I think, a little less than 7. I think it was around 7. I mean, it wasn't very acidic, but
What's the pH of the blackwater ecosystem?
the pH, I
Of
think
the
you
water.
said, of the water itself is 7, but
Yeah, in the blackwater.
in the black from like 4 to 5.
It's less than that, 3 to
3 to 4, so very acidic. So maybe the complex medium wasn't acidic enough for this bacteria to produce enough
4. We've done
metabolite.
it both ways.
Oh, you have?
Yeah, and there's really little difference with the more acidic.
Okay. So yeah, that was what
Yeah.
was my tell you in the brain.
I had definitely manipulated
The
pH.
acidity is mostly in water, not necessarily the soils surrounding water.
Right.
And as you said, we have that horizontal groundwater flow. Yes.
So did you go back and check because you said the THF was bioactive, so have you gone back and checked that, or?
Yeah, that's where we isolated
Okay,
these
yeah.
compounds from, because it was
Okay.
2 separate things, isolating the endurocidin, and then this THF fraction, which we found the unsaturated fatty acid to be. And that was
and that's
our,
why it wasn't a good.
yeah, it wasn't a good candidate.
And so
as an
it
antibiotic.
was the acetylnitrite, was that the other one?
Yes, we purified the endurocidin from the acetonitrile, and we first got this unsaturated fatty acid in our THF solution.
So is this something that, I mean, like what stage, like you're talking about like drug development,
Yes.
what stage is this for drug development?
So for drug development, I went from the beginning of just the fermentation of the bacteria all the way to the end to having this purified, characterized compound. So I did the full spectrum of drug discovery, which is what I found to be the most interesting about
Because
it.
your question is, what stages
Like
is it
what?
becoming a drug?
Right, so I mean, like, how close is it?
So yeah, I mean, if this was a good candidate and I had characterized it and I had purified it, then I could go on to maybe presenting it and hopefully getting.
Are you talking about fatty acids or peptide?
Both, actually.
From
I guess just
the,
in general.
from the endurocidin, I mean, we had our semi-pure endurocidin. We could modify that or go on and use that for whatever uses we want. So I mean, that's finalized. That would be a final antibiotic right there. But my THF fraction, this unsaturated fatty acid, we got it to the point of characterization, which then you can move on and push for saying, okay, we have this bioactivity, we have this compound.
Why isn't it a good candidate?
Why isn't it a good candidate?
Her question is, how are these, as far as coming along the road to becoming drugs?
Okay.
You kind of mentioned it before, but maybe you want to reiterate why fatty acid specifically is not. a good
Okay,
candidate.
why is not a good candidate? Because it acts as a detergent, as I was saying, and this is not something you want in your body. I mean, you wouldn't ingest soap to kill bacteria, correct? So, and that's exactly what this metabolite is, more of. But it's more stronger than a soap. As I said, it was keto conjugated, so that's why it gave us such strong inhibition. Yes.
Could it work as a surface bacterial killer? Would it be
Yeah.
an effective compound?
I mean, with the inhibition we got, it could be, but we were looking for a target to be just Staphylococcus aureus, and we wanted this to be in vivo and all that, so it was not good candidate. Yes.
So tell me what you think about the ethics of using this NRADN.
And
Concerning what you've now done, wasn't it NRADN the additive, the feed additive?
Androgen, yes, is the feed additive, and it's not used in the U.S. It's actually just used in South America and in Asia. Yes.
So
It's-
tell me what you think about the ethics of that.
So the ethics behind it is they're not just injecting these chickens full of this antibiotic. It's actually working as a hormonal messenger to tell other bacteria to back off because they're using such low concentrations in their poultry feed additive. As we said, as I was talking about earlier, it's really difficult to get the Streptomyces fungosidicus to produce the endurocidin. So there's a high demand and low production rate.
It doesn't move.
But is there an issue, yeah, you don't really answer.
sorry.
Is there an issue in using antibiotics in feed animals?
So increasing resistance. is that-
Specifically, this antibiotic, which you mentioned so far,
Mhm.
there is no resistance known for MRSA, so it seems like a really important compound. And they're jumping into chicken feed, and we know what happened with tetracycline in the US.
And caused all that resistance. Well, the nice thing is there has been no known mechanism of resistance to
Well, yeah.
the way it incorporates itself.
What'd you say?
I said yes.
Yeah, that's right.
Yes,
The
yeah.
question isn't
But hopefully
if,
not.
it's when.
Yeah, if or when this happens, but I mean, it's been almost 20, 25 years. 40 years that this has been in use, and still the resistance is really giving, I mean, it's pretty strong.
What's that telling you about how it works?
That it must not work in a pathway that they can just fend off. They can't just pump it out. I mean, it's incorporating what endurocidin does as a peptide is just get into the cell wall of the bacteria and lyse it.
So why is it used in the US?
Why is it not
I'm
used
just,
in
if
the
it's been
US?
used for, yeah, I'm just like, if it's been in use for 40 years, I'm surprised.
The FDA.
No,
well,
FDA doesn't have anything to do with it.
What
It's
organization?
an agricultural product.
Agriculture.
Right, USDA.
USDA.
That's who regulates feed additives.
Not good enough for the federal.
Well, FDA is only drugs.
Okay, so then USDA is for agricultural uses. Yeah, I mean, I don't really know why the US hasn't been using it. I'm guessing because of the whole resistance strain. Yeah, the whole concern, but it's been used for so long.
They more tightly regulate feed additives, but somebody has to apply to use it. So it's on the company a little bit. There's a lot of hurdles to get new feed additives.
Mhm. Exactly. A lot more stringent than South America or Asia.
So you also touched on difference between pumping an organism full of antibiotic or
Yeah.
putting it in a feed.
Mhm.
What are the implications of that?
Of putting in the feed rather than injecting
Yeah.
the. Okay.
What happens?
So what happens? Why they're putting in the feed? They just, as I said, low concentrations. They just want the hormonal activity of it being like a messenger for other bacteria to back off, rather than curing a chicken of its like let's say Staphylococcus aureus.
Do you remember what
infection.
we talked about, what the organism is that this controls in chickens?
Clostridium. It is targeting Clostridium
Perforangians.
perforangians.
That's a huge problem.
Injuries.
And that's an
The
embryo?
caries fungus,
Right,
could
caries.
you address caries?
It's a gram-positive bacteria. No flora.
So why doesn't Streptomyces fungicidatus or whatever it is, die from endocyte?
Why doesn't it die from endocyte? I mean, I'm hoping it doesn't die from what it produces.
Yeah, but why?
Why? You know, maybe because the cell wall of the Fungicidatus. I mean, as I said, they grow as kind of fungus-like, and maybe their mycelium isn't as
It will kill other
isn't as vulnerable. With the mycelium, isn't as vulnerable as other bacteria.
But I think the carrier is getting at something. Maybe we could take it up in the thing.
Okay. Another thing to talk about.
Oh, great, there's too many microbiologists in the room.
Yeah.
So sad.
Control now.
Anybody else?
Well, thank you guys again. Grab as much food as you can on your way out.
Keep eating that.
Yeah.